So in this section, we're going to go through DNA denaturation. So in the introduction areas of this unit, we've already talked about DNA denaturation. So I'm only going to briefly go through what that is, but specifically, when we take a look at DNA denaturation, it really has to do with, once again, coil versus helical states. So remember back when we were talking about proteins, we had our coil helix transition states, and we were also able to assign weights to the coil state and the helix state. We're going to do the exact same thing here, just so that you can get an appreciation of how certain models can be applied to different types of monomers for different macromolecules. So let's start off with an introduction to DNA denaturation. So as an overview, I simply want to draw a quick sketch of what denaturing DNA would look like. And so let's draw a model of DNA being denatured. So when DNA is denatured, originally we know that it has to be in a helical state. When it gets denatured, it's probably going to go into a coil state. So I'm going to draw something like this. Where this portion is in a helix state, similar to this portion. We then have an area here which is in a coil-like state. And I want to separate our analysis of this particular area because this is a loop of coil-like states. So when we're trying to model DNA being denatured, we obviously have to separate them into separate monomers. And those monomers can simply be bases. So why don't we separate these into bases? So I have my bases here. And they have to be paired up. So I have 1, 2, 3, 4, 5, 6, 7. So I have 7 bases at the top, matching with 7 bases at the bottom. And this represents a denatured portion of your DNA. So I want to mention here that each base can be in a coil, or helix state. So now looking at this, we should be quite familiar with it because we did the same thing for proteins when we looked at the amino acids. So now what we can do is say that for a coil-like state, we define C as a base pair in a coil state. And we define H as a base pair in a helix state. specifically here, I want to mention that in living cells, a loop or coil is induced by proteins that unzip DNA. So this is just a quick overview of a DNA unzipping model. So let's take a look at the next slide, just to compare what is base pairing versus base stacking. So let's just divide this screen in half. I want to go through what base pairing is. So it's just a brief introduction of different terminology that we use. When we take a look at base pairing, remember those two sets of bases that I drew in the top and bottom DNA strands? Now let's draw the two DNA strands here. And specifically, I might have a base on the top and bottom strands, and there's going to be hydrogen bonds that stabilize base pairing. So altogether, this would be called base pairing. Now, when we're taking a look at a model, we also have to think of how we can elongate or extend this base pairing, and that's basically called base stacking. So let's just draw the same thing again as best as we can. So I had this. So this would be a base pair or base pairing. And let's draw another pair here. I'll just say this is base pairing, but you're also connecting base pairs together to extend the chain. So this would be considered base stacking. So it's just different terminologies that you can use. Base pairing would be between your A, T, and your CGs. And then base stacking would just be an extension of those base pairs as you go along the DNA. So we've gone through an overview of DNA denaturation and some terminology with respect to base pairing and base stacking in terms of modeling. Now let's actually move on to analyzing the coil versus helix states. So we need to introduce weights for all states. We're going to say that the first H, or helix, is sigma S. We're going to say that the second H is S, or sigma. So once again, the explanation is very similar to before. We have sigma S, with sigma being very small because it's harder to form that first helical state. So taking a look at this, just doing a little bit of further analysis here. Sigma is much smaller than 1 because the first H state is always more difficult to achieve. Then we can actually take a look at the weight for HH. So what is the weight of HH? We can say that it's WHH is equal to sigma S times S is equal to sigma S squared. So this is just a brief exercise, and all we've really proven is that we can pretty much apply the same technique as we did for proteins. The only issue we have is in proteins, there's no such thing as these DNA bubbles or loops. So the exception we have to consider is something called DNA looping. So what exactly is DNA looping? We're going to take a look at that and introduce something called the zipper model for DNA. So in terms of looping statistics, let's do a quick diagram first of what I mean by this. So remember our initial diagram for DNA denaturation? I had some sort of bubble in this area, and this is considered a constrained loop. That is not completely random. So there is some order and structure to it. It technically is in a coil-like state, but it is ordered because it's in a loop. So its purpose is to pack DNA. And so taking a look at this, I want to kind of zoom in and let's say this could be a loop broken down into many different sections. So we call this J for each section. From different looping statistics and modeling that have been done, we can say that the probability of each state is going to be proportional to 1 over j to the power of 3 over 2, where it's been determined that the probability is approximately equal to 3 times the square root of 6 over pi n cubed. So you might be thinking, where is this even coming from? The most important thing that I'd want you to understand is that you would have 6 different configurations of a 3D lattice structure. Essentially 6 degrees of freedom. So taking a look at each of these nucleic acids, or the bases that we were talking about, consider each of them to be blocks, truly blocks with 6 different faces and 6 different configurations. And therefore, if you were to calculate the probability of a specific state, it would be the multiplicity of the looped structures over the total number of states. So let's take a closer look at this. I know just presenting the model is not enough. So the important number to remember from here is 6. We're thinking about blocks as our building blocks. Let's now move on to the zipper model. And when we take a look at the zipper model, we have our coil states, and we have our helix states. And let's say the sequence is given as CCC and some sort of H, and it continues on and on. If I were to calculate the weight of this, think about how many C's I have. I have 5 C's. Oops. I have 1 first H, and then I have n subsequent H's. So the weight itself for each of these coil states, remember there's going to be a base pair up here, or a base here, and a base below. Each one of these has 6 different configurations. So let's say 6. So that means if I'm taking a look at a sequence, I must remember it's a sequence of base pairs. So if I have a coil-like state, it means that I have 2 bases that are in a coil-like state in order to connect with each other, which means it's going to be 6 squared for each, and I have 5 C's with this sequence, so it's 6 squared 5 times. So that this corresponds to my 5 base pairs in coil-like states. So just as a recap, I want to mention there's 2 base pairs per C. So that means we have something like this. Okay, so the next thing, we have 1 first helix state. So it's pretty much always going to be one of those. So it's going to be times sigma s. So this would be our first H. And then we have n number of H's. So we have S times S times S times however many more S's there are. So we have n H's. So therefore the weight is going to be equal to 6 squared to the power of n minus n. And let's just consider this as, let's define this a little bit more closely. This should be n minus 1 H's because this is considered n equals to total number of H's. So we have n minus 1 because we've taken away the first H. So then we have 6 squared, capital N minus n for the number of H's, and that would give you the number of C's with the 6 squared probability or weight. Then we have sigma s to the power of n. So then we have an equation for the weight of n number of H's in our zipper model. So remember, with weights, we also looked at the partition function in our protein studies. So we can apply the same knowledge, and why don't we consider applying the partition function to this model. So I won't be showing all the work, but we get Zn is equal to the summation from n equals 1 to capital N of all the weights of n H's. Then it'll be equal to, we have 6 squared to the n minus n sigma s n. This is then equal to 6 squared n 6 squared negative n sigma s to the power of n. So what I can do is rewrite this as Zn is equal to 6 squared capital N sigma, which is just a constant, times the summation of s over 6 squared to the power of little n. So then we can use the partition function to quickly calculate the weights for specific sequences if we want. So expanding our knowledge from before as well, back when we looked at proteins, we applied the partition function to our polymers. We also were able to figure out the fractional helicity for proteins. So there's nothing stopping us from applying the same thing to the zipper model for DNA. We still have a polymer system. Just now, it's made up of monomers of nucleic acids versus monomers of amino acids. So let's take a look at fractional helicity. So using the sum of a geometric series, in which case we have this being a geometric series, if you remember from calculus. I won't be testing you on this ever, but that's where this comes from. We can then say that the partition function is equal to sigma 6 to the power of 2n. We get this huge term, s over 6 squared, 1 minus s over 6 squared to the power of n, divided by 1 minus s over 6 squared. So we get this huge term, and what we can do is apply similar techniques to those shown in previous chapters. And what you end up with is a fractional helicity term that is equal to 1 over n times the number of h's, and that becomes 1 over n. After we take partial derivatives, we have s di over di sigma of the partition function divided by Zn. So this is the equation for the fractional helicity of DNA in a denaturation or zipper model. So if we ever want to figure out how many what portion of the DNA is being denatured or is still in a helical state, we can apply this model to try and figure that out. So as a final step, what we want to do is take a look at the melting temperature So thinking back to before, we presented a graph of fractional helicity over some sort of temperature. Now we have this labeled as T over T room temperature. And you're going to get some sort of curve that looks like this. And this would be a fractional helicity of 1. And at some point you're going to get 0.5. And wherever 0.5 is, you're going to get some sort of Tm, or melting temperature, that is going to be approximately 1.4 times that of room temperature. So that's an example for DNA, and this is an example of a curve with 100 monomers. So this is just an example of different ways we can apply the models that we've created. So keep in mind, we've taken a look at a graphical representation of the DNA denaturation model. We've taken a look at how we consider base pairing versus base stacking. We've applied all of our knowledge from proteins to, in terms of coil helix states, along with partition functions and fractional helicity, to the DNA zipper model, and try to come up with different mathematical expressions to determine the statistical weights of different sequences. So it's expected that you would know how to use the same approach to figure out the total weight of a specific sequence.